To the folks that already dialed in, welcome to our Friday Coffee Chat series. I'm your host today, Dennis Koester. I'm the Director of Industry Research and Relations here at Drug Hunter. And we have Dennis Hu, our CEO, and Lew Pennington, our Director of Scientific Research, here today talking about unusual motives that are found in recent clinical candidates and in drugs. So let's maybe start it off. Dennis, what were some of the unusual motives that you've been seeing in recent clinical candidates that were kind of fascinating to you?
Oh man, there's so many. There's so many cool ones. So it's really hard to pick just a handful. So I'll try to keep this within time, I guess. Um, Maybe just to give some context, I can share my screen. Yeah, so one of the tools that's really popular with our members and that I personally use all the time is our structure search. So a lot of people don't know this, but we actually have all of the molecules of the month indexed by structure in the structure search tool, as well as all of the FDA approved drugs. So one of my favorite motifs that seems to be increasingly popular is the azetidine group. So what's interesting is if you look at all the approved drugs and you search for the substructure, what'll pop up is mostly beta lactams, right? So when people are thinking about using azetidines as an amine, that's not really what they're thinking about. But there are a handful of approved drugs. I think there's about 3 total where they have an azetidine amine in them. I think cobimetinib has an azetidine amide. Baricitinib has this interesting disubstituted azetidine sulfonamide. Um, so some interesting molecules. Uh, but that's why I like our molecules of the month database, because we index a lot more clinical and preclinical candidates from industry. There's only, like I said, 3 azetidine containing approved drugs. But if you look at the molecule database, there's actually quite a lot of advanced clinical candidates, oral clinical candidates, IV clinical candidates, et cetera, that do have azetidines, azetidine amides, et cetera. You even have macrocycles with azetidines in them, making a peptide bond, and spiromolecules, covalent molecules, some really cool stuff. So azetidines, I think a lot of people initially, are afraid of them because they might open an acid or things like that. I actually personally worked on this program where we did see a little bit of that in the synthesis, but they're actually quite stable, and you do see a lot of oral molecules. So if we click into this compound, for example, so this is FLX475. This is a first-in-class CCR4 candidate. This thing, even though it's got a relatively unstable looking N-aryl azetidine, it actually has a 72-hour human half-life and is orally bioavailable. So I think we can look up the oral bioavailability data. It's about 53% in rats. And if we scroll down here, I believe we can find the human dose as well. So yeah, in human dosing studies, it had dose-dependent oral bioavailability with strong PK/PD in humans. 75 mg PO/QD dose achieved the desired pharmacology. So something to consider. Azetidines can be a nice, I don't know what's the word for it, non-rotatable group, not a very conformationally labile or a floppy group. That can be a nice structural anchor or linker in a lot of molecules. Another nice example with an azetidine is Luna 18. So I'm just going to find that one with our text search. So Luna 18 has an interesting azetidine amino acid in here that I believe they use to help with conformational restriction of this macrocyclic peptide. This is a super cool molecule in and of itself. It's an oral macrocyclic peptide that is cell penetrant. And this is a phase 1 oral KRAS inhibitor, targets the KRAS SOS1 protein-protein interaction. So pretty cool story. I encourage everybody to take a look at that if you have time. Some other interesting motifs that I think might be interesting to this audience are acrylamides. So using the structure search, we can also take a look at how people are using acrylamides, both in approved drugs and in more recent industry clinical candidates. So I'm going to try drawing this here. And this will pull up a lot of covalent molecules, obviously. Some pretty famous ones like this KRAS molecule from Genentech, KRAS molecule from AstraZeneca, etc. Mirati's compound. Uh, and one of the ones that I think is really cool is this fluoroacrylamide from Mirati. So if we look at this one, we can find some articles on this. And I believe we have a case study on, let's see here, adagrasib versus sotorasib, where we talk about this extensively. So if you look at the fluoroacrylamide, what's really interesting about this is, as a chemist, you might expect that this more electron deficient acrylamide would be more reactive. But actually, if you compare adagrasib, a fluoroacrylamide containing compound, to sotorasib, you can see that in whole blood, this compound is a lot more stable than sotorasib. And this translates all the way through to higher higher dog, better dog PK, better dog bioavailability, lower clearance in higher species. So this is a puzzle for a lot of folks. Why would fluorinating an acrylamide make a compound more stable? And here you have a matched pair in this article where you can see with compound 18 on the left, this is a fully non-halogenated acrylamide. Adagrasib here on the right has this fluoroacrylamide. And again, you compare these two directly, you see the whole blood stability is way higher for the fluoroacrylamide. Dog PK is much better. Half-life is about 10 times longer for adagrasib. Why is that? So you can read this article to learn more about why that is. Jennifer Petter had a great explanation at ACS in Indianapolis. But if you're working on covalent molecules, this can be a nice way to brainstorm how people are building warheads. So speaking of that, I think if we search chloroacrylamide as well, there is an article on Vivid Ion's recent Keep1 engagers, where they have a really interesting cis substituted chloroacrylamide. And they find that this is much better than the trans one, and it's surprisingly, again, more stable than you might expect. There's a nice explanation in this article as well about why the cis isomer is less reactive. But another interesting motif that so far I haven't seen in any other drug candidates, but I'm sure people in the audience may know of some other historical ones. But super, super interesting example, really novel warhead that I hope gets used more often. I just have a handful of others. Do I still have time, Dennis? I could go on for days about this stuff. I just love this stuff.
I think you can still go on for like 5 minutes
5 more
or so. I
minutes?
mean, it's
Okay.
super interesting stuff. I mean, all the things that you're highlighting, I think people that work on covalent drugs, I mean, can really learn a lot about the warhead design here, right? Of what different ways there are. I mean, early on, people really started with the disulfide tethering strategy, right? And then cooled it down to acrylamides. And now you have so many more options to design covalent warheads. I think, yeah, you really nicely outlined that. But yeah, maybe you have a few other motives that you currently are following. That would be much appreciated.
Yeah, so speaking of covalent warheads, actually that reminds me, there was a really cool BMS candidate from a while ago that has a pyrimidine chloride that is actually a covalent warhead through an SNAR mechanism. So
Nice.
let's see here if this pulls it up. And actually, what's interesting about this is this motif is sometimes used in non-covalent molecules, reversible molecules, and I wonder how reactive these are in those cases. But this molecule, CC99677, so I guess it's more accurate to call it a Celgene compound. This is super cool because it's an example of a phase 2 oral candidate for immunology that doesn't have a acrylamide as a warhead, but actually has a pyrimidine chloride that reacts through nucleophilic aromatic substitution. So something to consider, both when designing halogenated pyrimidines and also when trying to find more drug-like covalent compounds. All right, one of my favorite covalent compounds, and this is going to blow everybody's minds. It's serpiglenistat. Uh, this compound is wild. It's a diazo compound that's an IVSC phase 1 or phase 2 candidate in advanced solid tumors. So this is pretty cool. It's a prodrug that has a diazo group. Again, that's something that, you know, anybody who's worked with diazo compounds in the lab knows that they're super reactive. You put them in methanol with a little bit of acid, they just, pop off. This thing is actually in clinical development, which is cool. So this is a super interesting mechanism for selectivity. They argue that this is selective for tumors because of this prodrug. In certain tumors, it'll hydrolyze through serine proteases to release an active metabolite, whereas in healthy GI tissues, a different set of esterases hydrolyzes it to an inactive metabolite. So one of the questions I think from the audience coming into this was, for each of these motifs, would you recommend using them or would you not recommend using them? I think of the ones that I've shown so far, azetidines are probably recommended. I think quite a few industry groups are using those. The fluoroacrylamide, increasingly, we're seeing those on programs. Chloroacrylamide also seems worthy to try. The diazo compound, seems maybe more like a one-off, but is pretty eye-opening to see that these things can be stable for some time in people. The last compound I have to show, just because Dennis is wearing a bow tie today, is if you search for cyclopropanes, I think you can find a bow tie compound in here somewhere. Maybe I should have searched for the spiro cyclopropane molecule. Let's see if I can find this. There is a Merck compound that targets LRRK2, where a bow tie, this molecule, I love this molecule. So Merck
Me
had this
too.
really cool, yeah. So this one's for you, Dennis.
Thank you.
Yeah, so this is super cool. I think they found that this was really unique in driving potency or combination of potency and properties, but this really nicely positions a nitrile in a place that I think displaces water molecule. I can't remember, but you can read this summary and take a look at this as well. Super, super cool, innovative group there. So that I'll pass it off to, I don't know, Lew, you want to go next?
Sure, I'll go next. So as Dennis alluded to earlier, we are having some Zoom issues. So Dennis is going to do the searches for me, and then I'll do the voiceover. This is not an issue with the drug hunter substructure search. It's an issue with Zoom that we'll be fixing in short order. So the first uncommon motif I'd like to talk about really revolves around the phosphorus atom. So the first motif is a phosphine oxide. This is a phosphorus atom, doubly bound to oxygen, and then 3 alkyl groups. So this is a fairly uncommon structural motif in drug design and discovery, but it is gaining some traction, and that largely comes from the success of brigatinib. So this is a drug, an approved drug that was discovered some years ago. And in fact, I know the person who did that. And, one thing today folks have to consider, and this sort of came up, I think, in some of the questions is, when should you reach for these sorts of things? And, honestly, as medicinal chemists, I think we all have learned that we need to be as opportunistic as possible. We need to follow the structure-activity relationship data. And really, we're only limited by our creativity and skill. So in the case of brigatinib, for example, somebody was creative enough to say, hey, why don't we do a phosphine oxide moiety to engage in a favorable interaction? And so the idea was there, but they couldn't figure out how to make it. And so they hired a really skilled medicinal chemist who came in and figured out how to make that molecule, which ultimately led to the clinically used drug brigatinib. So I'll just give her a shout out. That's Jan Romero, currently at Pfizer, a good friend of mine. So the molecule we're going to talk about today, it's not brigatinib. We're going to talk about SY5609. It too contains this phosphine oxide group, and it was designed intentionally, rationally, is a bioisosteer of a sulfone analog that had been explored in the program. And what they found in this program, looking for inhibitors of CDC7 kinase for oncology indications, is that when they swapped out this sulfone with a phosphine oxide, they saw that they got improved potency and metabolic stability. Now, other optimizations were required, after this initial bioisosteric functional group exchange, but ultimately that phosphine oxide made it into the molecule. Now, they didn't report an X-ray crystal structure of the molecule bound to CD7 kinase, but they did a lot of rigid docking and molecular dynamics simulations that suggest that phosphine oxide oxygen atom engages in an intramolecular hydrogen bond with that indole NH group that you see there in the molecule, and then also serves to engage in water-mediated hydrogen bonds with lysine 41 and aspartate 155 of CDK7. So really a interesting story. I would recommend people look into that. This is an older, very brief sort of summary of a molecule. One nice thing about Drug Hunter is if further information that you want isn't located in the Drug Hunter write-up, then you can always follow the links to get more information. So phosphine oxides, I think, are a really fascinating, underutilized structural motif in drug design. Another fairly uncommon phosphorus-containing motif are phosphinic acid groups. So this is a phosphorus atom doubly bound to oxygen, two alkyl groups, and then also a hydroxyl group. And in this case, the molecule that's really interesting to me is S62798 from Servier, shown here. This is an intravenous inhibitor of TAF1A, carboxypeptidase, and this is intended to be used as a blood clot degrading agent for, stroke and in other types of indications where blood clots are an issue. And so this molecule ultimately was terminated, but I thought I'd discuss it today because it is a very fascinating structure. And one interesting thing about this phosphinic acid group is that it binds to a zinc atom of TAF1A. And additionally, that cyclization that they have there, that imparts some conformational rigidity, and also serves to enhance the selectivity of the molecule. So really a fascinating and unusual structural motif that did the job it needed to do, but ultimately this molecule was terminated. So the next atom that I think is interesting, and this has been of interest for some time now, is boron. So the first boron-containing motif I'd like to talk about. This is a boron atom bound to an alkoxy group and a hydroxyl group. So if you just draw the substructure as one alkyl group bound to boron and two alkoxy groups, it'll lead to make one of those methoxys a hydroxy.
Oh, okay. Sorry about that.
Sorry about that, Dennis. I was a little unclear in my explanation.
No.
And also, too, folks, I do apologize. I'm actually suffering through my third bout of COVID, so my voice is a little hoarse and my brain's not quite working as well as it should. So bear with us. Yeah, there we go. And so, so looking at QPX7728, I think it's further toward the bottom. Yeah, this is an oral reversible covalent inhibitor of serine and zinc metallo-beta-lactamases for multi-drug resistant gram-negative bacterial infections. And this is intended to be an ultra-broad spectrum antibiotic medicine. So what's really interesting is you see a cyclopropane, you see it's a cyclic boronate. I mean, this is really a fascinating structure. And what they found is that the boron atom of QPX7728 interacts with an oxygen atom of the catalytic serine 70 and serine beta-lactamases. And interestingly, this boron atom in QPX7728 interacts with the oxygen atom of the catalytic water molecule in zinc metallo-beta-lactamases, and that alkoxy group off the boron atom actually engages in an interaction with the zinc atom of those zinc metallo-beta-lactamases. So really with this structural motif, you're actually engaging in two really interesting interactions, and they're different depending on the particular beta-lactamase that you're looking at. And so, I think boron is going to continue to be used more and more in drug discovery. We're now maybe 20 years into people using boron. I think it's established itself is a legitimate structural motif in drug design. I remember talking to a high-level person from Vertex many years ago, and this is during the early days of boron in drugs, and there was a potential opportunity to collaborate, and one person in the audience said, "Borons for morons." And so, I think that's a quote. I'm not gonna, I don't know who said it, but anyways, I think we've evolved since then, and I think people understand that, there's a time and a place for boron. So here we go. So the second molecule I'd like to talk about today that contains boron is M3258. This is an oral reversible covalent inhibitor of the immunoproteasome proteolytic subunit LMP7, and this is intended for multiple myeloma. So this is interesting because by targeting this subunit of the immunoproteasome, you can likely have greater or equal efficacy for multiple myeloma, but reduce toxicity, reduce side effects, because the immunoproteasome is only expressed in certain cell types, B and T cell types. It's not ubiquitously expressed as the constitutive proteasome is. So this is really a clever way of going about cell type selectivity. So I thought this was pretty fascinating. And in this case, the boron atom of M3258 interacts with the oxygen atom of threonine-1 in LMP7. So really a fascinating story here. And that's all I have. Boron and phosphorus, I've never personally used them in a drug discovery program, but gosh, I would love to someday for sure.
I mean, Lew, there's also a bortezomib, right? I think that has an approved drug for multiple myeloma that contains boron as a proteasome inhibitor too, right? So that's in my opinion also a very interesting example, as I have personally worked on proteasome inhibitors, not in the cancer setting, but more like in an anti-parasital setting.
Yeah.
Yeah.
I am proud to have made some phosphine oxides myself, and I was honestly shocked by how polar they are. I think a lot of times when you try to use them as an isoster, you're replacing pretty lipophilic sulfones or something like that, but they're like several log units more polar, which is interesting.
Yeah, so let's try with this, if the Zoom is going to work out. We had an audience question actually from Alex, and he was wondering about alkynes in drug discovery. Of course, that was actually what I was going to talk about. As an ex-Trost student, that is, of course, one of my favorite moieties, an alkyne, right? Because those are very easy to make, right? There's a lot of chemistry out there developed for alkynes. And, it's just, I think, a group that is increasingly popular in drug design. Now, Dennis and Lew, can you both see my screen okay? Or, yeah?
Yep.
Okay, perfect. So yeah, let's put this in, and Dennis had already showed you how we differentiate here our molecule database and our drug database. And I just wanted to search a few acetylines here in approved drugs, right? And what you will notice is that, people used acetylines in many, many cases, most often as covalent warheads. But what you'll also find is these steroidal drugs here, for example, danazol and others. And, I was really wondering why, and you scroll down here, you see more of these steroidal drugs that contain this terminal acetylene group here directly attached to make this alcohol a tertiary alcohol. So I was wondering, why is that? And I dug a little deeper into that. And it turns out that, the metabolic pathway here would be oxidation. If you wouldn't have that acetylene here, would be oxidation to the corresponding ketone, and then that would deactivate that particular steroid. So as to kind of protect this from metabolic oxidation and metabolic inactivation, this acetylene was found to be optimal. And really, many people, copied that same strategy even in the 2000s. So, this is a strategy that certainly worked. Now, terminal acetylenes, people tended to be kind of afraid of and shy away from for multiple reasons, right? I mean, there's CYP inhibition that people have been thinking about. There's, metabolic activation, TDI, for example, bioactivation. And the mechanisms for those are actually pretty interesting. They can actually be 1,2 rearrangement, and then you have a ketene, and then that ketene is attacked by GSH. So I thought the mechanism for that is incredibly fascinating. But the examples that I want to show you today, where I think acetylenes have been used really interestingly, is one, this compound here, this KRAS on inhibitor from Revolution Medicines, which I think is a really nice example of how to design a warhead. Again, the concept here is endlessly fascinating. So you really, this article is something you should be looking into. And again, they started with this sulfone tethering strategy, and it had actually acquired this compound here from Warp Drive Bio, who had found this concept. So with this acrylamide, of the stability of this compound, the whole blood stability just wasn't there, as you can kind of see in this example here. And what they did is they came up with this einamide warhead here. And this einamide warhead, that in this concept of of this KRAS inhibition by actually taking out KRAS irreversibly out of the equation with this new mechanism here, that einamide is actually way more reactive and compared to this acrylamide. And that's because there's a pre-organization of this molecule where the nitrogen binds in the pocket to pre-organize it for the attack of the cysteine. But of course, you also want to make sure that, now you have a more reactive warhead. So potentially your stability is going to suffer, your whole blood stability. And that's how they kind of monitored the stability. And one of the strategies to kind of stabilize your acetylene here, your einamide in this case, then is to introduce additional steric hindrance around that acetylene to then have really a compound that's way more stable. So I thought that was a very interesting use of stabilizing a covalent warhead, really elegant covalent warhead design. And this particular covalent warhead is something that I have not seen in other candidates before. Now, if you look at this particular compound here, of course, another thing that stands out is this strange heterocycle here, right? This is a 1,2 diazonane, a 1,2 diazonane. So that actually comes from the original discovery here of sangliferin. Sangliferin is a compound that was discovered in the Novartis library, and later Greg Verdeen figured out the mechanism of that compound. That's actually a cyclophilin and IMPDH2 binder. And yeah, that's a motif that I've been reasonably fascinated with, and that probably we're actually going to have a flash talk from Gil Overy that we'll hear more about, hydrazines in drug discovery. I think that's an endlessly fascinating story as well. Another story about the acetylenes that I want to briefly share is this compound is also a KRAS inhibitor. Is this KRAS inhibitor here from AstraZeneca? And really interesting, this motif, what they try to do is they try to optimize this compound for brain penetration. So the original compound that they discovered here had a really high efflux ratio in the MDCK assay. That's how they actually monitored it. And they're really trying to optimize for compounds that had lower efflux ratios. Now, for the potency, one of the things that they found when they ultimately moved away from this indazole here and moved to this phenol type here was that they had to use this oxazepam, the 7-membered ring here with the nitrogen and the oxygen, to pre-organize the conformation. I think that was a really fascinating use of also another maybe underutilized heterocycle, this oxazepam, also something that I haven't seen very often. But in terms of how the acetylene was ultimately used, so they tried a lot of groups here, including a nitro group, sorry, not a nitro group, a nitrile group, and then also the acetylene. And originally, they had nominated this compound here, the terminal acetylene, as the clinical candidate. And when they had done their GLP tox studies, they found out that after 7 days, after a longer period of time, they saw a muscle wasting toxicity both in dogs and rat, surprisingly not in mice. So they really dug into the cause for this. Why was it, why, did this muscle wasting tox occur? And they found that it was related to aldolase A inhibition. Now, they found that because aldolase A, as you all know from high school, in the glycolysis is an enzyme, and the substrate of it is fructose 1,6-biphosphate, and that really accumulated. So that's how you can tell that this enzyme was inhibited. So now they really analyzed that, and aldolase A, interestingly, is something that AstraZeneca didn't look into in their proteomic screening. They looked at a whole bunch of enzymes, but aldolase A was not among them. So, really interesting for everybody who's working on covalent warheads. Maybe aldolase A is something that you need to be adding to your cysteine proteomics. So now how they figured out that it was coming from this acetylene was they actually, looked into the structure of how this was going to be binding to aldolase A, and they found, that, there was a weak hydrogen bond with that somewhat acidic CH here, right? To asparagine, to an asparagine in the vicinity. And the design idea that solved this problem was just to create a steric clash there. So install a methyl group here in order to get rid of this aldolase A activity. And that's exactly what they did. And that's how they then ended up with their clinical candidate here, which also ended up being very nicely brain penetrant with a low with a low efflux ratio here. And they nicely really characterized this in PET studies as well. So yeah, acetylines, I think, are getting more and more popular. I mean, again, people, shied away from those originally, but again, you can also use them really interestingly as, because the click chemistry is very, very popular. So for labeling, PET labeling, and other interesting things to look at target engagement, for example, you can tag on fluorophores with acetylines because the chemistry is so easy. So I think that was just a very fascinating example for me. And I'll just leave it with that. Yeah, and I think we can probably go to audience questions from here. What do you think?
Yeah, sounds good. Yeah, nice picks.
Yeah, definitely. I enjoyed this too. Yeah, I learned a lot from both of you guys. Like really interesting stuff actually that you guys picked out. Yeah, so Anne is wondering, Anne Tran from Amgen is wondering, what is people's thoughts and experience in the increased presence of bromohydroarenes in recent drug approval? So this year there have been 2 approvals, both are oral. Aprocinitan and denicopan. Those are the 2 examples. What do you guys think? Bromohydroarenes.
Well, they're better halogen bond acceptors. Bromine's got a larger van der Waals radius than chlorine. So if you're projecting into a very tight hydrophobic binding pocket or sub pocket, maybe bromine will give you a little bit better van der Waals interactions, or maybe if you're actually engaging in a halogen bonding interaction, it'll be so much stronger than a chloro, and maybe if it's combined, then you get effects of both. Of the halogen atoms, nobody's afraid to use fluorine. We love fluorine. People are less afraid to use chlorine. I think there's really nice perspectives being written on the magic chloro atom, and how it can serve as a more perhaps metabolically stable lipophilic group versus a methyl group. But I think people start getting a little bit nervous when they move to bromo and in iodo analogs. And I think justifiably, and there are iodine containing drugs, but you have to start worrying about the chemical stability of iodine containing drugs. But like we said earlier in our comments, as medicinal chemists, we have to keep an eye on that target product profile. we have to understand, you know, the route of administration, the dosing schedule, the dose, whatever, and we just have to be opportunistic. And so, you know, my view is you go in with your eyes wide open, don't be dogmatic. And so part of the reason we have to build our knowledge base, part of the reason we want people to use Drug Hunter is so they're building their knowledge, people are aware. And if they move into these unusual motifs, they do so knowing the potential risks. But no, I'm pleased to see that, you know, with our discussion so far today, we're just seeing an explosion of diversity and novelty in drug design. I think it's a great thing.
Actually, I see Yoshi Ishihara is in the audience here. Maybe that's the topic for your next big review: bromine-containing molecules.
Yeah, we have Yoshi. Just I'm going to put in a plug here too for Yoshi. So we're going to have Yoshi too as a guest, and he will be talking about the magic of chlorine and also the magic methoxy group as well on the upcoming Flash Talk. So stay tuned for that. And yeah, Lou, I think you summarized it very well. I mean, there's bromine also in marine natural products, right? I mean, people, I think, were a little bit nervous about the redox activity of those methyls. Oh, sorry, no, I'm saying methyls of those halogens, right? Like especially iodine and bromine. But yeah, I totally agree with you. As long as the data doesn't stop you, right? You should be going forward. I mean, there's a lot of preconceptions, I guess, in medicinal chemistry. One of the things that I think we believe in at Drug Hunter is like you go for it. That only the data is going to tell you where to go and when to stop, right?
I think that, Dennis, that might be the first time I've heard about halogenated natural products since my PhD like 10 years ago. But actually,
I sound very academic. I'm sorry.
No, I mean, that was a big surprise. I remember looking into it. There are like thousands and thousands of brominated and chlorinated natural products, like algae and whatever. They're just spewing them out in the ocean. So yeah, I think bromine's not as scary of an atom as you might think.
Right. Well, maybe let's move on to a little bit scarier atoms here. Jeffrey Yang in the audience is wondering about selenium. Selenium in drugs. Now, selenium, that's something that I think of when I think of selenium, I think of shampoo. I think it's like in hair conditioner, it contains selenium. But yeah, in drugs, what do you guys think? I haven't seen any selenium containing drugs.
I'm trying to remember, you know, one of the that does remind me of a different topic though. I don't, Lou, do you know of any selenium containing clinical candidates? Actually, let me see. Let's search the selenium atom.
What about what's the Paul Ehrlich drug?
Oh yeah. That's right. The magic bullet. This is an anti. I think it's a hat drug, right, for human African trypanosomiasis. Epsilene. Yeah, somebody is saying it. Epsilene in the audience. Yeah, that's right. Epsilene. Wow. Yeah, I didn't remember that. Great that we have audience engagement. Thank you guys.
Yeah, no, so yeah, so I mean, selenium is we're starting to see more publications, you know, medicinal chemistry publications with selenium, but in terms of drug approvals, it's very few and far between compared to other things. But again, just like we've been saying, you know, it doesn't mean you don't do it. It means if you feel the data is directing you in that direction, then give it a try. But do it with your eyes wide open. You know, even nitro groups. There are lots of nitro group containing drugs out there, and it's all about the context of how you're trying to exploit that.
One of the interesting things there is here, I do want that reminded me of this article on Drug Hunter. Can you see my screen?
Yes.
Yeah, so we do have a poster of approved covalent drug mechanisms. And I remember one of the surprising things about this is we actually have a lot of enzymes that use selenocysteine. And actually, one of the reasons some metals are toxic is actually they inhibit selenocysteine containing enzymes. So we have selenium in our body and we need selenium. And actually, there are some drugs that are covalent inhibitors of selenocysteine motifs. So that's an interesting random tidbit, somewhat unrelated, but.
No, it's definitely related. Yoshi is actually replying in the audience that
Oh,
you're
really?
right on with that. He was just talking about
There we go.
selenocysteine.
Yes,
Yeah,
exactly. I love it. It's so interesting.
yeah, no, definitely. Yeah, other questions are any sulfur-6 containing drugs beyond sulfoxyms? Gagan is wondering about this.
Oh, well, let's search. I'm gonna punch in the sulfur atom on the structure search.
Yeah.
You know, I know there's been a lot of talk about using, so we're talking about like SF, hold on a second, let me
SF5,
read the question
maybe?
again. SF5 groups.
The question was how about, I think, sulfur-6 functional groups beyond sulfoxyms? Yeah, would that be SF5?
Yeah,
SF5, yeah, is
SF5.
something that,
Hmm.
yeah, that you can use as a very nice bioisoster for example, tert-butyl groups, right? It's like very sterically hindered, but not as, lipophilic. So if you want to maintain, your lipid, that's maybe something you can look at. I mean, sometimes it's really differentiating in the talks, surprisingly, from what I have seen.
Yeah, I
But
think
yeah,
yeah, that's probably another one of those ones where everybody's waiting for somebody to take the first step, but
yeah.
nobody wants to be first there. Um,
Synthesis wise, I think it's also opening up, right? It used to be like really difficult to make SF5 containing groups, arenes, because the chemistry is just so horrible with like fluorine gas and stuff like that. You need to reuse like really terrible conditions. But I think now more and more SF5 building blocks are becoming available. So it's a little easier for medicinal chemists to introduce them. But yeah, ultimately, if somebody discovers a drug, we probably have to figure out how to make these things on really large scale, and that could be another problem. Well, another question here is oxetanes and medchem. Do you guys have an opinion there? I mean, there are a lot of things.
So this is
Danuglipron,
this
for
is
example.
this is one I was very soured on oxetanes earlier in my career. I had tried it several times in drug discovery programs, and I got to the point where I was like, oh, I do not like oxetanes because every time I made one, they were chemically unstable. Uh, but I've been humbled, and I've learned through the work of other people that there are a lot of oxetane containing drugs out there in the clinic, and approved. And there are ways of designing oxetane drugs that are stable. And again, this gets to the point of context is everything. Not every atom or functional group is bad in every context. It's really very specific situations.
Yeah. No, for Occitane, yeah, so the most of the drugs, if you look into them, they are like, natural products that contain like highly substituted oxetanes. The drug candidates, so there are a lot of drug candidates that contain, oxetanes, for example, fenebrutinib, a BTK inhibitor that contains like an alpha unsubstituted oxetane. Then there's danuglipron, and danuglipron is a really interesting story. It's Pfizer's oral GLP-1 receptor agonist. They actually intentionally used this oxetane, and in their case, it gave them 100-fold potency boost because it was just the perfect group for the interaction with that GPCR GLP-1 receptor. So really, it gave them the right potency and also maintained that stability. They actually had a HERC issue earlier in the campaign. And one interesting strategy that they used is they introduced a carboxylic acid, and typically acids are not a liability for HERC. If you look at what kind of compounds are typically have a liability for HERC, it's like really basic lipophilic amines that sometimes non-classical HERC inhibitors are like molecules that have a certain shape. Y-shaped molecules are certainly liable for HERC inhibition. So yeah, that danuglipron campaign, I think, is a really nice example for a molecule that contains an oxetane that was really taken far into the clinic, unfortunately has been discontinued. But again, what Lew said, I 100% agree with that, right? until the data stops us. I think we shouldn't think in dogmas. It's all really context dependent. When do you need this particular oxetane? And it is a pretty powerful group. Like I think in phenobrutinib, it's basically on a piperazine. It's like a piperazine that is substituted with an oxetane. So like sometimes it's just really the perfect geometry for an interaction that you're seeking.
Actually, I think danuglipron is still in development with a once daily formulation. It was the
Oh,
twice
okay.
daily formulation that was discontinued, but I think it's still in phase 1. I could be wrong though.
No, I could be
haven't
wrong
checked on
about
it.
this. I defer to you, Dennis.
No. Yeah, definitely a lot more interesting clinical candidates. Actually, I remember speaking of, to both of your points, Lou, about the instability. One of the biggest surprises I had in the lab was one of my colleagues. So, for a lot of amines, before you do compound submission, you do Bach deprotections with HCl, right, to free up amines. And we had an oxetane containing molecule, and we found a side product that came from the HCl ring opening of the oxetane. And that ended up being a picomolar compound. It was like 1,000-fold more potent than the oxetane itself or any of the starting points. So, Lou, to your point about keeping an open mind, sometimes you find surprises like that might not be what you intended, but are pretty interesting. We didn't end up using oxetane, but it led us.
So one atom that hasn't come up yet is arsenic. So at Drug Hunter, we believe in getting our facts straight. So in Salvarsan, Paul Ehrlich's antibacterial drug, it was arsenic, not selenium.
Okay,
I had to
nice.
fact check myself. But yeah, even arsenic appears in drug molecules from over 100 years ago. So
And actually, triarsenic compounds are biologics, according to the FDA anyway.
right. Oh yeah, I remember your post on that. That's great.
Yeah. Maybe another question before we close. When considering unusual structural group, when does the lack of approved examples start to worry you that there's a fatal flaw versus they're just underutilized? So I think that's a pretty good question.
Well, certainly, you'd have to dig into whatever data is available. And if you still have a design hypothesis that strongly indicates you should maybe try that, but there's no precedent for it, and you can't find data that suggests you must stay away from this at all times, and I doubt there's anything out there like that, to be perfectly honest. Again, it's all about learn as much as you can, and then go forward knowing what potential risks there are, and design and execute the experiments that let you know if those risks you think are part of that functional group or that atom are actually the case in the context of your molecule and your program.
And that's one of the reasons we launched the Drug Hunter Molecules of the Month database and structure searches. If you look at the data set of approved drugs, it's actually very small. I mean, if you think of modern targeted, approved drugs, you only have a handful from 1990 on that are what we think of as like useful examples. But if you look at clinical candidates or preclinical candidates, there's a lot more, really advanced compounds that are good examples, perfectly safe, stable, achieve their target product profile, but maybe failed for, unrelated biological reasons or whatever, competitive reasons. So I think looking beyond the scope of just approved drugs as examples, but also looking at, sets like this can help.
Yeah. Maybe here's another question about alkenes. Alkenes as bioisosteres of alkynes and aromatics. Yeah, that's a good one. Alkenes. Does any example come to mind for both of you?
I've seen them in lots of but natural products, but have I seen them in drugs? I don't recall ever seeing that motif
Yeah.
in a drug.
Same. Certainly not in an approved drug, right? Maybe in some candidates. Yeah, not 100% sure. Maybe we'll get back to that. We'll get back to that question. All right. There's epoxides. What other things? Yeah, I think we
Yeah,
covered
plenty of
most
oxides.
epoxides. Yeah.
Yeah. So I'm looking at the webinar chat and I see Arcady Weisberg, I believe, was asking about the potential for phototoxicity associated with bromine containing drugs. That I'm not familiar with. Phototoxicity, I've never personally had an issue with that, but you know, that would be a particularly frustrating toxicity to have to deal with. And you know, I believe there are perspectives written on phototoxicity and how to get around that within the last few years in JMedChem, I believe. Um, but yeah, I mean, that's part of the multi-parameter optimization challenge. It's not just, you know, what's happening, you know, in systemic circulation in a person sitting in a dark room, but also potentially when they go to the beach and the sun impinges on their skin and they have drug concentrations in their skin. So yeah, it's something to think about.
Yeah. Yeah, I think it's also, you know, helpful to remember too that you know, it's the concentration of the molecules. I mean, iodine containing aromatic groups might not be popular, but you know, we have to remember that thyroid hormone, you know, we've all got you know, 100, I think microgram or 100 nanomolar quantities of that circulating in our blood all the time, right? Thyroxine, these are all uh, iodinated molecules. So,
Yeah. Yeah, for phototox, I mean, there's a lot of assays that one can use. I mean, of course, you can to a first approximation by absorption. You can look at the absorption spectrum and see if there are any red flags there. And then there are actually a lot of in vitro assays that you can use. Like there's a T3T NRU assay that you can use to monitor phototox. But yeah, it's not always telling, and typically the proof would be like in an in vivo assay, but an in vivo phototox assay is very, very expensive, I'll tell you that. Yeah, you want to make sure that ideally you're clean in your you know, in vitro assays, T3T NRU, for example.
I did see a comment in the chat about epoxides, or a couple comments about epoxides. Interesting. I just searched in our drug database. There are a lot more epoxides that have been approved than oxetanes, or even azetidines. So, pretty not uncommon.
That's surprising. Is that as a covalent warhead
Yeah, it
then?
looks like mostly covalent molecules.
Yeah.
Although no, I guess not all of them. Well, I'd have to double check some of these, but yeah, yeah.
Yeah, that's surprising. Maybe one more question. We're almost coming up on the hour. Maybe one more question and then we'll wrap it up. What are the parameters to consider for acceptance of a drug for the FDA? This is maybe in general, like a very general question, I guess.
Well, it's all about safety and efficacy. That's what the
Yeah.
FDA is looking at. And how you get the efficacy and how you get the safety doesn't really matter, but you you have to have both to be approved by the FDA. You've got to have a therapeutic index that is appropriate for the disease indication and the patient population that you're going into.
Yeah.
So who
All
knows?
right.
If you demonstrate that, you could have a diazo drug as well, I guess.
Yeah, that looks like to me like yeah, something that you would do a rhodium catalyzed cyclopropionation with. That looks like a perfect substrate for a rhodium catalyzed.
You know, you never know. I bet there's some hidden sneaky enzyme in the human body that does, you know, some crazy thing like that.
Right.
It doesn't
All
actually do
right.
though. Oh, sorry.
No, go ahead,
We can
Dennis.
wrap up. I was just gonna, didn't like Francis Arnold engineer or something like that? Like, don't they have some kind of synthetic enzyme that now does cyclopropionations with diazo compounds?
I think I've heard about that too. Was it
This is
Francis Arnold?
I don't know. I thought I saw this in the literature at some point. This was years ago, but I don't know. Somebody in the audience who's more caught up on biochemistry probably would know better.
Yeah. All right, everyone. I think this was super fun. Oh yeah, yeah. Yoshi knows about this. So you're right, Dennis. Yeah, so this was super fun to discuss here. I'll put one more plug again. We'll have Gil Overy giving us a flash talk later in September about unusual groups in medicinal chemistry. to address kind of biases in medchem as well, maybe as a follow-up discussion. I hope you found this session useful, and again, I hope that helped you understand how to use the structure search at Drug Hunter and what we are all about, what we're all thinking about here at Drug Hunter. If you have any questions, feel free to reach out to us anytime. We're here to answer your questions, and yeah, we're here for the community. And yeah, thank you everybody today for your time, and see you at the next event. Thank you.
All right, thanks for joining us.
Take care.
Thank you guys. Bye.
Bye.